Hello friends, welcome back to my channel Plant Pathology Pathsala. So in this video we will discuss about isolation and purification of viruses, that is the plant viruses. So first we will discuss why the isolation and purification is needed, then about detail about the extraction, and finally we will discuss about the purification. What are the steps, why we need the purification, what are the chemicals that are used, what is the functional role of the chemicals. So each and everything in detail we will use, and I will also discuss what question can be asked in MCQ based exams like ASRB net as well as SRF exams. So in detail we will discuss. So without further delay, let's start the video. So first, the why isolation and purification is needed for viruses. We are not doing isolation and purification for the fungi. We just what we do for virus, bacteria or fungus, we inoculate that on artificial media. We just let it grow on that artificial media and then we do the purification step. But here in the virus we have to do the extraction. Why? Because virus is a obligate intracellular parasite. It need a living host for its growth as well as multiplication or replication. That's why we can't grow it on the artificial media. It needs to be extracted from the infected plant tissue. That's why the isolation as well as the extraction step is there. Why this extraction and purification is needed? Because for the identification of viruses, for morphological characterization, for physiological studies like physiological characteristic studies of virus. You might have heard about longevity in vitro, then the dilution endpoint. So physiochemical characteristics are there for that is specific for specific viruses. To study that also we need purified virus sample. Then about to study the infectivity. Okay, so we need isolation and purification for identification, characterization, physiological study as well as infectivity study. So let's discuss isolation and purification, various steps included in that in detail. First, about the extraction or the isolation. So this for the viruses actually we specifically use the extraction term, not the isolation. Why? Because we are extracting the virus particles from the plant tissue. What is the purpose of extraction? Purpose is to release the virus particles from infected plant tissue. Virus is an obligate intracellular parasite. So virus will remain inside a plant cell. So we have to crush that cell or tissue to release the virus particles. Then to maintain the virus infectivity during extraction, how we maintain it? By using some chemicals like buffers. Proper extraction buffer we have to prepare to maintain its infectivity or stability. Then prepare a uniform extract for purification. So extract the virus crude extract that we got after extraction of the plant extraction from the plant sample, that is most important, because if you are not getting good uniform extract then it will create difficulties in the purification step. So prepare a uniform extract. So purpose of extraction of plant virus is, first, most important, it will release the virus particle from the infected plant tissue. Then to maintain the infectivity during the extraction by using specific chemicals like extraction buffer. So we will discuss in detail what are the chemicals used in the extraction buffer, why it is used, what is its means characteristics. Okay, everything we will discuss in detail. Then the ultimate purpose is to prepare a uniform extract that will be used for the purification process, so that we will get good virus yield. And the principle of extraction of virus. So what is the principle? So virus particles are released mechanically by disrupting the infected tissue in a suitable extraction buffer while protecting them from the degradation. So the principle behind this is, we will mechanically disrupt the infected tissue. We will crush the infected cell as well as tissue, so that the virus particle multiplying inside that infected tissue will be released outside, and it should be done in a proper condition, proper environmental as well as chemical. Proper extraction buffer we will use, so that the plant material should not degrade the virus, especially the nucleases. As you all know that virus is a nucleoprotein structure, so the nucleic acid can be degraded by the nucleases present in the plant. So we have to protect that using extraction buffer. The final outcome of extraction is the crude virus containing extract ready for filtration or the purification. So outcome of extraction is, you will get crude virus extract, not purified virus. This is the crude virus extract you will get, that need to be that means you need to do the further purification step to get the final purified virus. So this is about the extraction, basic about the extraction process. Next, the extraction process. So like other extraction process, like extraction of DNA, RNA, so you have to first get the sample. So sample should be young, should be fresh. Okay, so select a fresh infected plant tissue, wash it, dry blot, then cut it into small pieces. Then you have to grind it, means you have to do the mechanical crushing. Okay, you have to crush that. So for mechanical extraction of the virus, you have to use the chilled mortar and pestle. This is the mortar and pestle. So using the mortar pestle, you have to grind it using cold extraction buffer. See, the condition here is, the mortar pestle you are using, it should be chilled. Means what you will do, you will keep the mortar pestle in the freezer, and just before extraction you will remove that, so that mortar pestle will be in chilled condition. And the extraction buffer you are using to crush that sample also chilled. Okay, so both the chilled mortar pestle as well as chilled extraction buffer you will use. Then you will do the grinding. Okay, the first is the infected plant tissue you will get, properly clean it, cut it, then grind it with the cold extraction buffer. Then whatever you will get, then you have to filter the grinded or homogenate through a muslin cloth or cheese cloth. So after the filtration, you will get the crude virus extract. Okay, so this is the extraction process. By the extraction process, what is the main purpose? You will get the crude virus extract. The virus is now out of the plant tissue. Okay, that needs to be further purification is needed. Okay, so main thing you have to keep in mind is, maintain everything in proper temperature condition, like this 0 to 4 degrees Celsius. Always keep the material, whatever material you are using, keep the material as well as the buffer, the extraction buffer, in 0 to 4 degrees Celsius. Why it is needed? To maintain the virus stability, because if you do not maintain the temperature, then the virus will be degraded. Okay, you will not get the good yield as well as good quality, and virus infectivity also lose. So proper temperature maintenance is important. This is about the procedure of the extraction of plant virus. Next, about the extraction buffer. Why extraction buffer is needed? To protect the plant virus particles from degradation and help in efficient release. So the extraction buffer name itself suggests that it will help in extraction, means virus is there inside the plant cell, so it will degrade the plant cell wall or membrane and make the virus available. Okay, it will release the virus from the plant cell. And second thing, the buffer means it will maintain a proper condition so that the virus particle will remain stable, it will not degrade. Okay, so this is the extraction buffer means. So what are the components used for the extraction of plant viruses? For extraction of different things, like if you are extracting the protein, or if you are extracting DNA or RNA, different buffer or different component you have to use. For specifically the plant virus extraction, these are the components. Why this component are used, we will discuss. Okay, what is the function of this chemical components? So first very important component is the phosphate buffer. So phosphate buffer, why it is used? Because it maintains pH. Buffer means definitely it will maintain the pH. Then another thing is it ensure the proper stability of virus particles. And the pH of the phosphate buffer should be alkaline. If the virus pH is alkaline, then actually it enhances the virus stability. Okay, so remember this point. The pH should be 6.8 to 7.4. Mostly 0.1 molar phosphate buffer is used. This can be asked in MCQ based question like ASRB net as well as the ASRB exam, that which buffer is mostly used for the extraction of plant viruses. That is phosphate buffer, mostly at alkaline pH. So alkaline phosphate buffer, it enhances the virus yield, maintain pH and ensure stability of virus particles. Next, the second most important component of extraction buffer for the virus is beta mercaptoethanol. This is a reducing agent. Reducing agent means it will donate the electron to the to various components like the viral proteins, and thereby it prevents the oxidation. It protects the infectivity by protecting the viral proteins. Okay, this is very important. Beta mercaptoethanol. What it is? It is a reducing agent. Okay, next is PBP, polyvinyl pyrrolidone. What it does? It removes the phenolic compound. It binds and removes the phenolic compound, and by this it prevents the browning as well as oxidation. If the sample will remain fresh, good virus quality or quantity you will get. So we have to remove the phenolic compounds, because phenolic compound will degrade the plant tissue and ultimately causing browning as well as oxidation. So to prevent that, PVP, polyvinyl pyrrolidine, is used. Next, the fourth component, EDTA. This is the most commonly used chemical in the extraction buffer for various things like DNA, RNA, protein, everywhere this EDTA is used. Why? This is because it is a chelating agent. It chelates the metal ion. So it chelates the metal ion and it prevents the metal ion to get attached with the various enzymes. You know, for activation of the enzyme or for proper activity, the enzyme need cofactor, and the cofactors are the metal ions only, this calcium and magnesium mostly. So when this cofactor will bind to the enzymes like nucleases or other proteases, then only that enzyme will be activated, and further it will degrade that particular compound. Okay, if EDTA is used, then it will chelate the metal ion. It make the aggregation, it form aggregation of metal ions, and that metal ion will be now not available for the nucleases or other enzymes. Then ultimately this activity of nucleases as well as other degradative enzyme will be not there, it will be inhibited. So this EDTA, this chelating agent, inhibit the activity of degradative enzymes like nucleases. See, virus is nucleoprotein. It contains nucleic acid. For the infectivity of virus, nucleic acid, DNA or RNA, is important. If the crude extract, the plant extract contain nucleases, definitely it will degrade the viral nucleic acid. So to inhibit the activity of nucleases, this EDTA is used. So now, uh, now it's your turn to mention the full form of EDTA in the comment section. So you have to write it in the comment section. What is the full form of EDTA? Next is the most important, ascorbic acid or sodium sulfide. So why it is used? It is an antioxidant. Definitely antioxidant means it prevent and protect virus from oxidation. Actually this ascorbic acid, this was asked in SRF 2022 or 23 I think, that means in assertion reasoning type question it was asked that why this ascorbic acid is used. Is it a antioxidant or it is a purifying agent? Like that it was asked actually. So and this ascorbic acid is a antioxidant. It prevent the virus or protect the virus from oxidation. So these are the component of extraction buffer: the phosphate buffer, then the beta mercaptoethanol, PBP, EDTA, ascorbic acid. So why this extraction buffer is needed? To maintain the virus integrity, improve extraction efficiency, and ensure virus maximum recovery. Okay, this is very important one. Remember all the points in MCQ as well as long time. Everywhere this is asked. Okay, in interviews also this is mostly asked question. What is EDTA? What is the full form of EDTA? Then what is the role of EDTA? If you will just tell that EDTA is chelating agent, how it inhibited the nucleases or other degradative enzyme? These are generally mostly asked questions. Okay, you have to remember this. Next is the very important step, precautions during extraction. Why precaution you have to take? So you have to take the precaution to get high virus recovery, to maintain virus infectivity and integrity, to reduce the contamination and impurities, as well as to improve the efficiency of downstream purification, because after extraction you have to do the purification also. So if you get the good crude extract, then definitely you will get good purified amount of virus also, with good virus yield, good infectivity, as well as without any contamination or impurities. So what are the precautions you should keep in mind? So firstly, the use of fresh young infected tissue. This is the basic thing. Okay, whenever you are using anything, means whenever you are doing any extraction process, the sample should be fresh young infected tissue for getting the better virus yield. Then keep the equipment as well as the buffer in chilled environment, that is 0 to 4 degrees. This is the most important step. Then what are the precautions during grinding? So grind the sample very gently. Don't means overheat the mortar pestle. Okay, avoid foaming also. Do the things very quickly, because if you are doing the things, or you are processing the sample quickly, then it will minimize the enzyme activity, because for enzyme activity proper timing is needed. If you are not giving that much time, you will skip the activity of the degradative enzyme. Okay, and the final is the most important one. Use correct buffer to tissue ratio, that is 1 is to 2 to 1 is to 5 weight by volume, depending on the viruses. Whenever we are going to any molecular laboratory, standardization of the procedure is most important. See, in every lab they are using different quantity of buffer as well as sample, so you have to standardize it. It may differ from virus to virus, and as well as condition to condition. Okay, so you have to standardize the buffer to tissue ratio. So this is all about the extraction process, the first isolation or extraction of plant viruses. In the next video we will discuss about the virus purification, the clarification, then about the partial purification and final purification. If I will discuss this also in the same video, it will become very lengthy. So in this video we will discuss only this much only, the virus, about the virus extraction, why it is needed, what are the steps, the homogenization and the filtration to get the crude virus extract, and the chemicals that is used for virus extraction are phosphate buffer, then beta mercaptoethanol, PVP, EDTA, ascorbic acid. These are very very important, remember this. And finally the precaution during extraction. Okay, so this is all about the extraction. Thank you for watching.